Welcome, everybody. Thank you for taking the time and coming into our webinar with the subject of the whole plant physiological identification and quantification of disease progression. I'm Karen Moshelion, to those who don't know me. I'm the manager of Plant-Ditech. Our company develops and sells the plant array phenotyping system. I will explain more about it later on. I will co-lead this webinar together with Shani Friedman. Let me just share the slides. Okay, I hope you see it well. Sorry. So let's start with a short introduction. Shani, who will lead together with me the webinar, she's a PhD student in the Faculty of Agriculture at the Hebrew University of Jerusalem. Her research focuses on the physiological responses of plants to biotic stresses. Shani previously participated in the Amirim Plant Science Excellence Program and won an Excellence Honor Scholarship. Her research back then focused on combining thermal imaging and physiological methods. I will explain a little bit how the webinar will look like. So we will start with a short introduction of the plant array technology and solution, and then we will move on very shortly after me. We will move on to Shani, who will share with us about her research of early disease detection and quantification, and a paper that she's just now in the process of publishing. We will then move to Q&A session. You're very welcome during the sessions, the talks, to put your questions into the Q&A, into the chat, sorry. And we will go at the Q&A session, we will go over these questions at the chat, and we'll try to answer most of them. So I just want to remind everybody that we are recording the session. So if you can keep the webcam and microphone off. And again, if you have any question, just post it in the Q&A, in the chat again, in the chat, so we can answer it later on. Okay, so let's start with the introduction of the plant array technology. The plant array platform is a high-throughput physiological phenotyping system controlling and measuring continuously and simultaneously many plants. It's kind of like a smartwatch, which is measuring the plant dynamic response, but for many plants simultaneously. And what we are doing, we are measuring, first we are controlling the soil and water conditions. We can control each and every plant differently, and with the feedback, also with the feedback loop with the measurement. So with some of the ways that we can control the plants are with also the measurements that we're taking. And the measurements we're taking are physiology parameters, different kinds of physiological parameters, together with the measurement of the environmental changes. We measure the plants about 500 times a day, all of them at more or less the same time. And all of this data goes into an analytical tool, which is measuring the plants in real time. So you can be at your home, your office, and look at your experiment via the data and see how your plants are doing, including statistics. You can see here the parameters that we're measuring. So transpiration, stomatal conductance, biomass gain, water use efficiency, root influx with the help of soil sensors, and also NDVR and IR with additional sensors, and atmospheric sensing together with soil sensing and even reservoir sensing. All of this is simultaneously and for all the plants at the same time. And this high-resolution data goes into the SPAC Analytic software, which is a cloud-based software where you can log in and see the experiment which is going on. With this system, you can benefit from deep insights thanks to the high resolution of measurements, so deep insight into the plant environment interactions. And also, we measure all the plants within days. Sometimes we can see the responses within days and sometimes weeks, but definitely less than full seasons. You don't need to leave the plant for the whole season, so it's a fast screening phenotyping system. What is the difference of this system versus other systems that are mostly the high throughput systems are imaging? We're quite unique in this space because we are measuring the function of the plant rather than the morphology or the structure of the plant or color of the plant. So it's a direct measurement of the functionality of the plant. It's dynamic throughout the day and simultaneous to all plants. And we can also control the conditions of the plants in many different ways. Very, again, similar to a smartwatch, which is measuring the physiological response of the body. We do it to plants. It's different from morphology, which is slower. It's only a few plants at a time, few data points, very complex analysis because it's indirect measurement and quite limited controllability. So these are the benefits of our system. You can see here one of our customers, Wageningen University, and mainly how the system looks in large scale. The main applications for our system is genetic screening, abiotic stress studies, nutrients and bio screening. These were the main use cases so far, and now we also added recently the biotic stress, seed treatments and growth practices. Biotic stress, you will see more in depth during the talk of Shani. The system works with many different plants. We have two sizes of the system, which are quite flexible by themselves. So we can work with plants, very small plants like Arabidopsis, through field crops and vegetables, until even trees, as you can see here in the picture. The system was developed at the Hebrew University initially, and we are a spin-off. The company Plant-Ditech is a spin-off from their development. It's commercial since 2017. We have so far over 60 scientific articles published based on the system. And you can see some of our customers here. Sorry, Wageningen that you saw before. Also Göttingen University, Korea. So these are just examples of universities. There are many more. Florida University in the US. And also from the food section, an example is Mars Wrigley doing cocoa plant screening under drought mainly. And Proform for the biostimulants screening. So these are just examples. We have different customers from these sectors. So this is for my part. It was short. It was just to introduce to those of you that don't know the system yet, to the system, to the principles of the system. Again, if you have questions, just post it in the chat and we will address them later at the Q&A. So before I refer to Shani, just as a reminder, please keep your cameras and mics off because we are recording the session. So from here, I just then transfer now to Shani. I will stop sharing and she will share from her computer. Thank you for listening.
I will open everything, just a minute. Okay. Hi, I will present to you a part of my research using the plant array to identify and quantify disease progression. Let's see. Okay, my name is Shani. I'm a PhD student at the Hebrew University in the Department of Plant Science. I've always had a deep love for plants, but I have to admit, I've unintentionally become quite good at killing them in all sorts of ways. Funny enough, this knack of causing stress to plants actually become the focus of my PhD. Let me introduce you to one of the top 10 fungal pathogens, Fusarium oxysporum. This is a soil-borne fungus, specializes in attacking tomato plants, causing a disease we call Fusarium wilt. The infection starts at the roots and moves up in the xylem, where it blocks water flow. This leads to wilting, yellowing leaves, and death. Fusarium oxysporum can devastate tomato yields, impacting both commercial farms and home gardens. So how do farmers manage it? One common method is using resistant tomatoes. We'll dive into that a bit later. How do we assess Fusarium wilt disease? I will explain by an example, by a picture from my work. This is 2 weeks ago. I inoculated my tomato plants with Fusarium. It's important to mention that all the Fusarium work we did is a collaboration with Dr. Shaikova from the Department of Phytopathology. For the first few days, we didn't see much. This picture was taken 11 days after inoculation, where you can see some yellowing and some leaves not fully open. As a scientist, you need to quantify this tiredness and yellowing. And how do you do that? Looking at the literature, there are two main ways to assess diseases. Usually, an experimenter visually inspects the plants and gives them a score. 0 means no symptoms, and 1 to 3 indicates various degrees of yellowing. However, this method can be inaccurate since it's subjective. I've also seen some articles discuss this issue. The other main method is measuring yield or biomass at the end of the experiment. However, I feel this misses some of the disease dynamics because it's in the end of the experiments. We want to offer a third option, using the plant array to assess the disease. By definition, disease is an impairment of the normal state of a plant that interrupts or modifies its vital functions. We have a tool that can continuously measure vital physiological parameters. These parameters like biomass, transpiration, water use efficiency, stomatic conductance, and everything else Karen showed. It's easy to use. It's quantitative, meaning not objective. It's objective, not subjective. Comparative, we can have infected and control plants simultaneously, and it's continuously. From day one, we can follow the disease. Our goals are to use the continuous physiological measurements to detect diseases in plants early, even before visual symptoms appear. Additionally, we aim to use the system to quantitatively assess the severity of the disease. Before I dive into the experiments we did and which Fusarium strain we used and tomato cultivar, I want to present you this overview. In all the experiments, we followed after the transpiration, the plant net weight, and visual symptoms. This table is a summary of the symptoms hierarchy we found in our experiments. In the table is when t-test showed the infected group is significantly different from the control. In all the experiments, transpiration was the first one to differ. Next was plant net weight, and lastly was visual symptoms appeared. So just from this overview, you can see that the plant array system provides early detection before visual symptoms appeared. Let's talk about plant susceptibility. People usually talk in 4 words: immune, resistant, tolerant, and susceptible. The problem of some of these words are not black and white. How tolerant is a plant? How susceptible is he? We suggest in talking in precise percentage of yield. Let's give an example. This is an industrial cultivar, M82, that is tolerant to Fusarium. And this is R13, which is susceptible to the pathogen. Following the plant weight after the inoculation, we could see the differences of the cultivars. You could see the trend starting very early on, but we would look at this stage. M82 control treated plant gets to around 220 grams. When inoculated, it loses about 15%. Interestingly, R13, the susceptible cultivar, produces higher biomass, 23% more than M82. In brackets, I would say this is not significantly higher, but when inoculated, R13 plants decrease biomass in 60%. There is a high correlation between biomass and yield. So now that we have these percentiles, a farmer could decide if he wants to take a risk and perhaps gain 23% and grow R13, or he knows he has Fusarium in his field and he is satisfied in losing 15%. It's quantitative numbers and it's early detection already very early on. We can see the trend. We did this with two other cultivars, Marmande Verte, MV, a susceptible cultivar, and Mutele, MT, a resistant cultivar. Following transpiration, here too, the resistant cultivar MT in gray is acting nicely. When infected, it loses some transpiration. The susceptible cultivar doing better when uninfected, but loses much more when inoculated. I can present this graph in one other way, the relative transpiration from the daily maximum, meaning taking the relative from the maximum plant to transpire the most. I found this presentation very interesting because you can see nicely the stabilization of the percentiles and perhaps how the system can early on quantify the differences between the cultivar's susceptibility. Next, I want to talk about quantifying fungal virulence. This is an experiment using two Fusarium strains. This one is a commonly used lab strain we called MVF. And this one is one we got from a seed company that's called virulent Fusarium because we got it from a contaminated field. We did a traditional seedling assay proving the differences between the strains. In a traditional assay, you take tiny seedlings, you inoculate them, and 3 weeks later, you score the symptoms. You can see that the virulent Fusarium inoculated plants were dead or wilted. And the MVF inoculated plants showed also wilting, disordered, or some were healthy but had browning in the vessels, indicating that Fusarium grew inside. This worked very well. You can see the difference between the two strains. And it's presented in this graph. From my experience, it is hard to decide sometimes what group a plant is in. And to find the browning in the tiny vessels is also pretty hard. But anyways, it works well. And I wanted to take it a next level, more field-like, with fertilization and sunlight and big pots. Using the plant array system, as you can see, in greenhouse conditions, after 18 days, the plants still look fine. Looking at the transpiration at day 18, here, we could find differences between the control and the virulent Fusarium. After 22 days, we found differences between all three treatments. This showing nicely how the disease dynamics and how it progresses. And the best thing about this is that it is before the visual symptoms. The visual symptoms occurred only after day 22. I want you all to notice that in greenhouse conditions, sometimes the plant don't die so quickly. It takes more time. And the last part of my talk, I want to shortly introduce you to 3 other plant pathogen interactions that were monitored using the plant array system. The first is Phytophthora infestans that causes the late blade in potatoes. Looking at the E is transpiration normalized to weight 3 days after infection, inoculation here. We could see significant differences between the groups. But you can see in the pictures, the disease is not yet significant until later. Wexler also used the system with peppers that had some kind of root rot. By the end of the experiment, the shoots looked quite similar. However, the roots showed varying degrees of rot. By following daily transpiration, you can nicely differentiate between the groups at day 19. Interestingly, they compared these findings with reflectance measurements across different stages of the root rot. I find this interesting and applicable to the field. I believe the first step is to fully understand how infection disrupts whole plant physiology through continuous parameters. And then by placing imaging systems over the plant array, we can identify how these physiological indices impact thermal and reflectance measurements. And this is an important step towards better understanding field sensors and drones. This work was done by Mishra. They used tomato yellow leaf crow virus. Healthy plants and well-watered transpire nicely up to 800 grams per day. But already from day almost 1, virus-infected plants transpired much less. Interestingly, they tested how virus infection impacts drought tolerance. When exposed to drought, virus-infected tomatoes are more resilient than uninfected tomatoes. You can see that they're getting to the critical point later. In the picture, you can see that the healthy plants subjected to drought are suffering significantly, while the virus-infected plants are still standing strong. To conclude, the lysimeter system enables early detection of diseases even before visual symptoms appear. Transpiration is the most sensitive physiological trait we can monitor, and this continuous system is ideal for quantitative comparisons between plants and fungi and may be utilized for breeding disease resistance in plants. These days, our work is in peer review, and we would like to hear your questions and feedback.
Okay, thank you, Shani. This is great. Thank you so much. And I do see some questions that appears, and I do invite others also to post their questions. So I think we will go one by one. I see a question from Carolina Lopez. Hello, Shani. How do you prepare the inoculum fusarium for infection, and how do you apply and also, and how do you apply it? And also, at what crop stage did you apply the inoculum?
Okay, so we used 3 weeks old seedlings. We cut the roots and then we put them in a suspension of fusarium and we plant them directly on the system. So there's a concentration and protocol we do, and it's very common used.
Okay, thank you. Next question from NoGap. On what kind of plant disease do you think this system works well?
We went on diseases that incorporate the vascular system because we thought it would be the best for the plant array system because it works on water traits relations. However, we saw it also in Phytophthora and also in like viruses, and I think pretty much it can work on everything because it follows the physiological traits.
Fantastic, thank you. I see another question from Tobias. Hi, Tobias. Good afternoon, Shani. Did you make use of pollinators in your experiments, such as bumblebees? For my company's experience, there is no pollen production in fusarium infected tomato plants. It might be interesting for you to have a look at this aspect too. Do you want to relate to that?
It sounds interesting. We did not get to the, in most experiments, we did not get to this to the stage of fruit, but definitely I can think about it.
Okay, great. Thank you. Yeah, Tobias gave you an idea.
Yeah. I didn't know that.
Fantastic. Again, from NoGap, what future research directions do you plan to pursue based on your current results?
We have currently an experiment using additional strains of fusarium, like one endophytic without that doesn't do any disease, and one that regained the disease we got from a Martin rep. Want to see if the system can differentiate between these two strains. And we might go to other plant pathogen interactions.
Okay, great. Next question, Rejane. Hi, Rejane. Would the system work to evaluate effectiveness of materials with the fungicide activity?
I'm sure, yes. I'm sure. I had an experiment we did, and the pepper one I showed. I think the pepper one I showed. They used fungicides, and they saw how it helped and how the plants grew stronger and transpired more after the fungicides. But it took some time.
Thank you. Dr. Desai, transpiration is affected by drought as well as wilt. How will you distinguish in the field from this or that?
You mean drought and wilt disease?
Yeah, if it comes, if I understand correctly, if it comes in combination with also drought as well as wilt. Yeah.
This is a good question, and this is like the focus of my PhD. I want to see if I can see differences between transpiration, like in the daily trend between like different kinds of stresses.
I can add to that with the system, you can either isolate different stresses or combine stresses. You can provide also together any kind of stress disease coming from disease or apply drought at the same time. There is actually a paper that we have on our website dealing with that, a certain virus, and Shani showed it before, and together with drought and how it actually helps in drought. Okay. Next question from Erkan. Thank you very much. Such a great presentation. I think seed treatment is scope of your study. Can you give us some detailed information about it?
What treatment? I missed it. I missed the word.
Seed treatment. Is seed treatment is the scope of your study?
We did not try seed treatment. We tried only like to inoculate them with suspension of conidias. Yeah.
So we have some experience with users that provided seed treatments on the system and applied drought stress, and they could learn the response of the plant to the drought stress thanks to the seed treatment and how this can help. So it can be done, and it was done already several times. Noga, again, a question: what are the benefits of the system versus visual methods, in your opinion, Shanique?
The system is very precise and quantitative. That's what I talked about a little bit. And in my experience, also visual inspection makes you come every day to the greenhouse, or every 2 days, depends how much you want it. But I think it also makes it easier, like to follow the disease.
Thank you. So following the disease and also being updated about what's going on in the experiment. If you allow me, I will also add that it helps to early detect diseases because of the high resolution measurement. So there is then the traits that we're measuring. So it's immediately analyze the stress when it happens, and also to quantify and tell me if this is what you experienced too. Did it help you to quantify the difference of responses by the plants between the plants?
Yeah, that's what I showed in this presentation, that I could quantify the susceptibility of plants and the virulence of the fusarium. I think it's also possible in visual inspection, but it's harder. It's not accurate. I think this method is much better.
Great, thank you. Erkan is asking again, which kinds of statistical programs did you use for your study? Was it hard to manage data and read them?
Because we're working with the plant array system and has a spec analysis, so everything comes directly to my computer, and all the data is like very easy to send to Jump. I use Jump and Nova tests. Pretty easy.
Right. What kinds of statistical programs did you use?
Jump.
In
Jump
terms
is a
of program.
statistical.
Okay, no, I thought he meant what kind of statistical methods, but he asked about programs. Okay. Dr. Herman is asking about, do you plan to measure secondary metabolites to see how drought improves tolerance to infections?
Interesting. Hi, Herman. Interesting. We did not subject drought in my experiments, only one I showed of somebody else's. We might see metabolites and how drought influences infections also in the future of my PhD.
We have some work like that done on in other areas that, for example, on cannabis plants, where our users looked for secondary metabolites, and so we have some examples like that. You can write to us, and we can share with you. Sanjay is asking, hi Sanjay, drought experiment and disease both in the same time of experiment, how to calculate the transpiration by your method? They apply together.
They apply together. I think you just need a control for each, like a control for disease by itself and disease with and without drought.
So you're saying
what I would
that
do.
you just have to separate the treatments, and by that you can identify the different transpiration behaviors from the plant. Yes?
I think so, yes.
I think so too. Okay. Dr. Desai, hi Shani. We see some diseases predisposed due to drought, heat, stress. How will your system help them? Predisposed, this means?
Drought, heat, stress.
Yeah.
I'm not trying to understand the question. Like, you're asking how drought, stress influences diseases?
So maybe Dr. Desai, if you can come up from in your voice, just telling us exactly what you meant.
Yeah. Good evening. It's a good lecture, I would say. Thank you very much.
Thank you.
We had one phenomics system also here in India established by, and there we used to work on these things. The thing is that whenever I apply some drought stress or heat stress, because I worked with wilt, the wilt suddenly increases at abnormally high rates. That's a predisposition. Say, for instance, if I have the climate change scenario, if I am taking it, the early part of the chickpea wilt is because of the fusarium, and as soon as soil temperatures shoot up, it will switch to root rot because of Rhizoctonia solani. So how would your system adjust to these kind of changes with which exactly the correct remedial measure is applied in the field? That's my point.
Can you differentiate between the different diseases and like do only heat, only the fusarium and only the other disease you talked about?
But in the field, they don't appear separately, is it not? They come together, and whenever they are together, either there is some kind of synergy between these two biotic and abiotic stresses. There is a synergy. When there is a drought, the dry root rot is more as compared to normal conditions.
So
Well,
actually, this is, sorry, Shani, that I jumped in, and you're welcome to add to what I'm saying, but this is the benefit of using such a system as ours, because you can provide the two stresses together and then measure the plant response to these two combined stresses. So you can really understand what's going on with the plant, when it's actually starting to react, how much is reacting versus, let's say, without drought or with drought. So you can apply the different treatments and see the differences. Shani, and if you have any.
And additionally, I want to say, in our greenhouse, I don't know if it's all greenhouses, we also have like sensors of temperature and humidity and light, so we can know like when the disease is enhancing more fast or slower by the environmental factors. So I have 10 experiments, and I could say at this temperature the disease came much earlier, and this temperature it came much later. So I can compare that.
yeah.
Yes, so in every system we actually supply atmospheric sensing, so
Yeah.
we can do that.
Yeah, interesting. Interesting. But then only the point is that you are not, neither you are directly measuring the drought stress, nor you are directly measuring the wilt. You are measuring the transpiration, which is a byproduct of both of them.
Yes, we're measuring the plant.
That's the whole point. Okay, good.
Unless you provide the treatment as only wilt without the drought, and then of course you can see what is the difference between the treatment. Okay, let's move on.
Thank you very much. All good luck for you, Shani.
Thank you.
Thank you. So next, it's more of a comment. Nada Kasem saying, thank you. It is a very informative presentation. Thank you, Nada. Next question is by Bulo Debesa. Sorry if I'm not pronouncing it exactly. How did you screen genotypes against the disease? Did you screen genotypes under disease hotspots or artificial infection?
The screen genotypes. I just got seeds that were told to be susceptible or tolerant to fusarium. I did not do any screening in my work.
You didn't check it with different genotypes, but you got basically from the same. It should have been more or less the same family.
They're all Leucopersky. That's a question. They're all Leucopersky.
Okay. Next question, Orna. Hi, Orna.
May I
Questions
a
of
little
him.
bit,
Yeah.
Bob's this point, expand a little bit. His point is probably the artificial infection means when you are doing it under glasshouse conditions, whereas hotspots are those where the wilt continuously comes year on year, because there is a lot of inoculum buildup by natural means. So these are the two different conditions, because the hotspot is a field condition, whereas artificial infection is under glasshouse conditions. So this is the difference between them. And they will throw differently two different diverse or dichotomous scenarios.
So what's your offer to use in the greenhouse, like inoculated soil?
I would prefer hotspot location, because that's one which gives much closer to the field scenarios as compared to the laboratory test. Because when I used to inoculate my chickpea plants with my highly susceptible, most virulent strain, it used to collapse between 9 to 11 days. Whereas if I give slightly different conditions, that collapsing extends up to 18 days. So that might not be a good measure if I am doing it. But then if you're, say, like if you take the field phenomics where you can do the hotspot locations directly screening part of it, I think probably you can get much better information and more close to the field conditions. That's my take.
Interesting.
Okay.
Okay, thank you. We continue to Orna's question. How do you see the use of the platform in seed companies? Shani, do you want to try and answer that?
You can answer that.
Okay. So I don't think we have a firm answer because we have tried it more from a research point of view, but I do see the potential in terms of better understanding of the how the disease is developing, when it actually starts to appear and reflect to the plant. Because in most of what Shani's work was done, and also in other experiments that involve biotic stress, we saw the phenomena only very late in the process, several weeks later. And in the data, it already started very, very early. So if it was really violent, it was even within days. So we could really follow to see how the disease is developing. And then you can see the differences between different genetics, how this is developing, and also so it's quantified, really the impact on the plant performance. So this is how I see it, Shani.
I also want to add that we talked with some seed company employees, and they told us like, wow, this is a cool system because we have diseases that we have to check like a whole season to see how they impact the plant. I don't remember which diseases he told me, but if this can enhance the research and save us time, this is in seed companies, it's going to mean money. But I'm not sure in every disease, transpiration or plant weight would be efficient.
Yeah, I think
Can
you're
I add
right. It depends on the disease, if it impacts the roots or the stomata. There are much better chances that we will see it in the data. If it's other diseases, we have to check. Yes,
Correct.
sorry, somebody. Yeah.
Can I add it too? I think there are two most valuable applications in seed industries. Quite a few of these Physaria are seed-borne up to 2%. So such plants can be eliminated if they can be detected early, so that the seed production programs are infection-free. That's one application. The second one is whenever they are doing some breeding programs, like nowadays hybrid pulses, hybrid oils, etc., when they are producing, the hybrid seed production, early detection will give you an advantage as to whether they're slow wilters or late wilters, and based on that, because late wilting is also one of the desirable traits. So these kind of traits can be characterized, with which the seed production programs can go much better. These are the two good applications I would see.
Perfect. Thank you so much. And I think we have no other questions at this point. So I think it's time also to say goodbye. And those that want to reach out to us and learn more and ask questions still after this session are very welcome to contact us. We have in the chat our details, how to get in contact. And also we can refer to Shani if needed to further discussion. And I thank you for the time for you to spend with us. And Shani, special thanks to you. You did a great job. And thank you so much for sharing with us your work and your hard work, and also the results, which are very, very interesting. So thank you so much. Thank you all.
Thank you. Thank you for the opportunity.
Okay. Thank
Bye-bye.
you very much. Good luck. Good luck once again.
Thank you. Bye-bye.
Bye-bye.